Hello there. My name is Tania Rodriguez from the class of 2022. And today I'm gonna go over the major takeaways from the alcohol metabolism small group activity. And this is my cat. Studying from the GI chapter in First Aid, our favorite system. Let's start with patient 1. A 21-year-old female presents to you with concerns about her predisposition for alcohol use disorder. In order to better address her concerns, we have to consider 3 questions. Number 1. What is the principal route for the metabolism of ethanol and which tissues are involved? Number 2. What is the microsomal oxidizing system, MEOS, or as I like to call it, MEOS, and how is it used to metabolize ethanol? Lastly, which genetic polymorphisms are responsible for a predisposition to or protection from alcohol use disorder? Has a heritability been established? What kinds of studies have been done on ethnic groups upon consumption of alcohol? By answering these questions, we will be covering LOs 2 and 3 for the session. Let's start with the first question. What is the principal route for the metabolism of ethanol and which tissues are involved? When we drink, alcohol is absorbed by our GI system and then is primarily metabolized by the liver. The first step in this process is the oxidation of ethanol to acetaldehyde by the enzyme alcohol dehydrogenase. And by doing so, a molecule of NAD+ is reduced to NADH. The generation of NADH has a lot of downstream consequences that we will cover later in this video. Acetaldehyde then is later metabolized into acetate. A reaction catalyzed by the enzyme acetaldehyde dehydrogenase. This reaction also generates a molecule of NADH from NAD+. Again, very important. Remember how I mentioned the first step in this reaction occurs in the cytosol? Well, the second reaction, the conversion into acetate, occurs in the mitochondria. Now I'm gonna go ahead and clear out my screen to make the next step easier to visualize. After the generation of acetate, most of it enters the bloodstream, as does some of the acetaldehyde. Both of them are then taken up by peripheral tissues, including skeletal muscle. In skeletal muscle, acetate is further metabolized into acetyl-CoA by the enzyme acetyl-CoA synthase. Does acetyl-CoA sound familiar? What is a very important cycle acetyl-CoA fits into? There's multiple right answers, but the one that I was looking for, the TCA cycle. As you may already know from biochemistry, the TCA cycle produces NADH, FADH, and ATP. Based on this, you may think that the generation of acetyl-CoA from alcohol leads to the generation of energy. And you're not wrong. However, remember how I mentioned the production of NADH in the earlier steps of this process was important? Well, when we consume a lot of alcohol, we generate a lot of NADH. This increased NADH to NAD+ ratio inhibits the TCA cycle. Consequently, the generation of energy through this cycle and oxidative phosphorylation is also inhibited. Lastly, how many of you have gotten a hangover from drinking too much alcohol? I know I have. The culprit? Acetaldehyde. Acetaldehyde is toxic and responsible for some of the alcohol-related damage seen in body tissues, especially the unpleasant short-term effects, such as flushing, headaches, and nausea. And that's it for question 1. Feel free to take a break before we move on into the MEOS, or as I like to call it, MEOS. What is the microsomal oxidizing system, MEOS, and how is it used to metabolize ethanol? The MEOS is an alternative pathway for alcohol metabolism. It normally metabolizes a small amount of ethanol, specifically 10 to 20%. However, it becomes really important when we drink a lot, constantly. Just like drinking what we drink at the Met Gala, but every day. This pathway also occurs in the liver, and it is CYP450 dependent, specifically CYP2E1. CYP2E1 has a higher Km than the alcohol dehydrogenase enzymes, which means it does have a lower affinity for ethanol. However, at higher concentrations of alcohol, more ethanol is metabolized through this pathway. This pathway also generates acetaldehyde and acetate, just like the principal pathway we discussed before. However, there are some key differences. Instead of generating NADH, this pathway is consuming NADPH. During this reaction, NADPH is oxidized into NADP+. When this happens, free electrons from NADPH are transferred into oxygen. This transfer of free electrons sometimes leads to the generation of free radicals, which, as you probably already know, are very damaging to tissues. In this case, liver tissue. To make matters worse, the expression of CYP2E1 is ethanol-induced. So the more we drink, the more CYP2E1 is expressed, and consequently, more free radicals are generated, leading to liver injury. Lastly, what's another pathway in which NADPH is very important? That's right, the HMP shunt, or pentose phosphate pathway. They're the same thing. This pathway generates glutathione, which protects red blood cells from oxidative damage. When NADPH is depleted, as it is in high consumptions of alcohol, red blood cells are no longer protected, and this leads to even more tissue damage. This is all you should know about the MEOS. Feel free to take a break before we move on. And finally, we've made it to our last question for this case. Which genetic polymorphisms are responsible for a predisposition to or protection from alcohol use disorder? Has a heritability been established? What kinds of studies have been done on ethnic groups upon consumption of alcohol? I realize this is just not one question. It's actually 3 in 1. And to better understand the whole picture, we should answer question 2 first. Has heritability been established? The answer is yes. Alcohol use disorder is thought to be a multifactorial genetically influenced disorder. And believe it or not, it all goes back to biochemistry. Remember those enzymes we talked about on question 1? Alcohol dehydrogenase and acetaldehyde dehydrogenase? Well, different people have different genetic polymorphisms that result in different enzyme isoforms for these 2 enzymes. You could have different alcohol dehydrogenase isoforms, and different acetaldehyde dehydrogenase isoforms. These isoforms specifically are protective against alcohol use disorder and are very common in East Asian populations and also 20% of the Jewish population. To understand why they are protective, we have to go back to the reactions from question 1. To review, when we drink alcohol, ethanol is metabolized into acetaldehyde in the liver by the enzyme alcohol dehydrogenase. Then the acetaldehyde is metabolized into acetate in the mitochondria through the enzyme acetaldehyde dehydrogenase. These specific isoforms have very high activity, and consequently, people who have them produce a lot of acetaldehyde. Meanwhile, this acetaldehyde dehydrogenase isoform has very low activity, which further contributes to the accumulation of acetaldehyde. I want you to ask yourself, why might an accumulation of acetaldehyde lead to protection from alcohol use disorder? Think back to the effects of acetaldehyde in the body. That's right, acetaldehyde equals bad. Acetaldehyde is the culprit, the thing that makes you hangover, headaches, nausea, flushing is all bad. So it makes these people not want to drink alcohol. And as a result, they are protected against developing a use disorder. In contrast, enzyme polymorphisms in which acetaldehyde dehydrogenase activity is increased leads to a predisposition to alcohol use disorder. Why might that be? That's right, it all goes back to acetaldehyde. If you have an enzyme that's getting rid of acetaldehyde faster, then what happens? You don't get a hangover. You don't get the flushing. You don't get the headache. You don't get the nausea. You don't get all these bad symptoms that make alcohol bad. And instead, you get all the good things, all the good feelings of alcohol. To answer the last question, there are some studies that link variant forms of the dopamine receptors, the neuropeptide Y gene, the GABA A receptor genes, and the serotonin transporter promoter with alcohol use disorder in different groups, but nothing's definite at this point, so don't worry too much about it. However, make sure you know how alcohol dehydrogenase and acetaldehyde dehydrogenase can protect or lead to alcohol use disorder. So what do we tell our worried patient? Well, it depends. Does her family have a history of really bad hangovers? Or vice versa? Does she have a family history of alcohol use disorder? Has she noticed that she doesn't get any side effects when she drinks, such as flushing, headaches, or nausea? Depending on her answer to these questions, you can advise the patient on her predisposition for alcohol use disorder. If she does have a family history and she notices that she doesn't really get hangover symptoms, then maybe, yes, she does have a higher predisposition for alcohol use disorder, and you should advise her accordingly. That's it for patient 1. Please go to the next video to learn more about patient 2 and the major takeaways from the case. Otherwise, thank you! And I hope you keep enjoying GI as much as I did.